Hello everyone, today I am going to explain the identification of presence of lipid or fat in a given sample in the physiological experiment of zoology. So here in the test we have to identify the lipid in the given sample. As we know, the 4 beakers are there. In that 4 samples, any one of the sample contain lipids. We have to identify in which sample the lipid is present. So we have to write aim, principle, procedure and result. Aim is the same like as question. To identify, to detect the presence the lipids in the sample. Principle means here the lipid is non-polar compound. So it is soluble in the organic solvents, that is ethanol. So then we can mix the sample with the help of ethanol and we can dissolve in that, and then we can add water. Then the end of the mixture, it will give the emulsion. The drop like white color emulsion is appear. That sample is give the presence of the lipid. If not, remaining samples cannot give the any type of emulsion with the help of adding of ethanol to the sample. So by the emulsion we can identify. In the sample, then only we can write A, B, C, D samples. For example, A sample having the emulsions observed. Here there is no emulsion occur. Then in the sample A, the lipids are present. So for the purpose, we can take a test tube, add sample and add ethanol. Then we can shake it. Then we can add water. Then emulsion will occur. Identification of fat or lipid in a given sample. So here we have a 4 type of samples A, B, C, D. So any one of the sample having the fat or lipid. So we have to take all samples into the respective A, B, C, D test tubes. But here we are just testing from the B sample. We have to take the sample into the test tube. And for the identification of fat or lipid, we can add ethanol. Why? Because this fat or lipid is dissolved in the ethanol and it can reaction and the fat or lipid is dissolved in the ethanol. Then we can shake and mix well. Then after the total droplets are dissolved in the ethanol of the fat or lipid, then we can add the distilled water to the mixture. Then it will form a white color emulsions in the test tube. So by that observation we can identify in the B sample the fat or lipid is presence. So here the lipid or fat dissolved in the ethanol and the droplets are dissolved. Then we can add distilled water to the solution and the white color emulsion will be appear. So by the presence of the emulsion we can identify in that sample we have a fat. So the operators are test tubes, droppers, ethanol and water. Thank you.